All right, so we're diving into biological classification today. Sounds complicated.
Yeah, it can be. Kind of like organizing a massive library, but instead of books, we're talking about every single species on Earth.
You sent over Chapter 2 from a biotextbook, which focuses on that. So where do we even start?
Well, like with most things, we start at the beginning.
Okay, back to the basics. I see the textbook mentions Aristotle.
Yeah, back in ancient Greece, he had some pretty simple classifications.
Like trees, shrubs, herbs for plants.
Exactly. And for animals, it was all about whether they had red blood or not.
Simple enough, I guess, but probably not that accurate.
Well, it worked for a while.
Until microscopes came along and blew everything up.
Pretty much. That's where Linnaeus came in with the two-kingdom system. Plants and animals.
Ah, that makes sense. Much more organized. But then what about bacteria?
Yeah, they didn't really fit. Fungi, too. The discovery of prokaryotic cells was a big deal. No nucleus, remember?
Right. So that's where Whitaker's 5 kingdoms come in.
Bingo. 1969, he kind of revolutionized things.
5 kingdoms. That's Monera, Protista, Fungi, Plantae, and Animalia.
You got it.
Yeah.
Whitaker looked at cell structure, how organisms get their food, how they reproduce, all that.
So much more than just what they look like.
Exactly. And it's still widely used, although there's the 3-domain system, too. But that's a story for another time.
Maybe a future deep dive. But okay, let's stick with the 5 for now. Starting with Monera.
You got it. The fascinating world of bacteria, they're everywhere, and I mean everywhere. Soil, oceans, even some pretty extreme places.
Yeah, the book mentioned that. Hot springs, even acidic mine drainage.
Crazy.
But they're essential for life as we know it.
Absolutely. They're like the recyclers of the planet, breaking down dead stuff, making nutrients available.
Oh, right. Like carbon, nitrogen, phosphorus, that kind of thing.
Exactly. And of course, they're super important for human health.
Both good and bad, though, right?
Yeah. Digestion, vitamins, even protection from bad microbes. But some bacteria, well, they're not so friendly. Cholera, tuberculosis.
Diseases, right. So understanding them is pretty important.
Absolutely. Now, the textbook mentions they're categorized by shape.
Coccus, Bacillus, Vibrio, and Spirillum. All those weird shapes. Does that actually matter?
Oh, it definitely does. Shape dictates function, even at the microscopic level.
Like, round shapes are good at resisting drying out.
That's one example. Rod shapes are great for attaching to surfaces.
And what about those spiral-shaped ones?
Ah, those are often found in bacteria that move through thicker fluids, like the bacteria that causes syphilis.
Oh, wow. So even their shape tells a story.
You got it. And it's not just shape. Bacteria have incredible metabolic diversity. Some make their own food.
Like plants with photosynthesis.
Exactly. But some even use inorganic compounds, something called chemosynthesis.
Like cyanobacteria. I love those. Blue-green algae, right. They were essential for oxygenating the early Earth's atmosphere.
Precisely. But most bacteria, they're heterotrophic. They get their energy from other organisms, like dead organic matter.
So like the decomposers we talked about.
Yep. And don't forget, nitrogen-fixing bacteria, they convert nitrogen from the air into a form plants can use.
Wow, they're like tiny little farmers. But hold on, what about mycoplasma? The textbook mentioned those. They don't even have a cell wall.
They're the rebels of the bacterial world. And yeah, no cell wall does make them vulnerable.
I can imagine. But there must be some advantage, right?
Oh, absolutely. It allows them to squeeze into tiny spaces and take on different shapes, too.
Sneaky little things.
Some are harmless, but others, well, they can cause infections. Pneumonia, urinary tract infections.
Oh, no fun. But even within this one kingdom, the diversity is mind-blowing.
It really is. Shows how evolution shapes life in incredible ways. And speaking of incredible, have you heard of Protista?
Oh yeah, the catch-all kingdom. I read about that. It's like the miscellaneous bin of eukaryotic organisms.
That's one way to put it. Mostly single-celled, a few multicellular exceptions, but all eukaryotes.
Meaning they have a nucleus and other organelles, right?
Right. But beyond that, it's a real mixed bag.
The textbook mentioned that even defining this kingdom can be a challenge. Like, is it a protist or a plant? Who decides?
It's definitely tricky. Some protists share characteristics with plants, animals, or even fungi, but they don't quite fit in anywhere.
So they're like the evolutionary crossroads.
Exactly, showcasing the amazing diversity of eukaryotic life. But I think we should probably dive into those fascinating groups within Protista after a quick break.
Sounds good to me. We'll be back in a flash to explore the world of chrysophytes, dinoflagellates, and more. Don't go anywhere.
Back to the fascinating world of, well, life, I guess. This time, fungi.
They always seemed kind of mysterious to me. Important, but, like, hidden.
Definitely essential. They play huge roles in ecosystems and, yeah, often overlooked.
So not plants then?
Nope, totally separate kingdom.
Yeah.
With unique characteristics like how they get their nutrition.
Right. Heterotrophic. They
Yeah.
can't make their own food like plants.
Yeah.
They absorb stuff from their surroundings.
Exactly, which is what makes them such great decomposers, breaking down all that dead stuff.
Nature's cleanup crew.
Huh, that's one way to put it. Keeps those essential elements circulating, for other organisms.
Makes sense. But the book also mentioned they form partnerships, not just breaking things down.
You mean the symbiosis stuff? Fascinating relationships, really.
Like lichens. I always see them on hikes, growing on rocks and trees. They look kind of alien.
They're pretty unique. A partnership between a fungus and either algae or cyanobacteria.
2-for-1 deal.
Exactly. Fungus provides the structure, the protection, and the algae or cyanobacteria, they make the food. Photosynthesis, right?
A tiny ecosystem.
Yeah.
Cool. And what about mycorrhiza? The book talked about those too, with plant roots.
Those are essential. The fungal hyphae, those spread-like structures, they basically extend the reach of the roots, more surface area.
So the plants can get more water and nutrients.
Exactly. And the plant, well, it provides the fungus with sugars from photosynthesis.
So everybody wins.
Exactly. Symbiosis at its finest. Now, speaking of those hyphae, that's actually how most fungi are structured. Long threads forming a network called mycelium.
That's all about the network.
And they can spread incredibly fast, colonize new areas, find food. Super efficient.
I read about that giant honey mushroom in Oregon. Spans, like, thousands of acres. Biggest organism on Earth.
It's mind-boggling, right? All connected underground. But now, let's talk about how these things reproduce.
I remember the book talked about both sexual and asexual reproduction. Asexual seems simple enough. Just break off a piece, you've got a new fungus.
Fragmentation, yep. They can also bud, like grow a new individual right off the parent, or use spores.
Spores.
Tiny cells that get released, float around, and if they land in the right spot, bam, new fungus.
Clever. But sexual reproduction in fungi seemed a bit more complicated. The book mentioned plasmogamy, karyogamy,
Yeah.
meiosis.
It is a bit more involved. Plasmogamy is basically the fusion of the cytoplasm from two compatible fungi.
So they're, like, getting close.
Exactly. Then karyogamy, that's when the nuclei fuse. Creates a diploid zygote.
Okay, and meiosis.
That's the division part. Reduces the number of chromosomes back down by half. Produces haploid spores.
New generation. But there's also that Dicarian stage, right, in some fungi.
Good catch. That's where those two nuclei from the different mating types hang out together in a single cell before fusing.
Like a trial run.
You could say that.
Yeah.
But okay, enough about reproduction for now. Let's jump into those four main classes of fungi. We've got phycomycetes.
The water-loving ones here.
Well, often found in aquatic habitats, yeah. Some of the most primitive types, a septate hyphae, meaning they lack those dividing walls.
So more like one giant cell.
In a way, yeah. They reproduce asexually with things like zoospores.
Zoospores.
They're motile, can swim around, or they use non-motile ones called aplanospores, and sexually, it's all about zygospores.
Okay, try to keep all this straight.
Don't worry, it's a lot to take in. But think of examples like mucor, rhizopus.
The bread mold culprits.
You got it. And albugo, which is a parasite that causes white rust disease in plants.
Okay, what's next? Ascomycetes.
Yep. Sac fungi, mostly multicellular branched septate hyphae this time.
Septate meaning they have those dividing walls?
Exactly. And they reproduce asexually through conidia and sexually with ascospores. Those are formed in special sacs called asci.
I remember that. Asci. In fruiting bodies called ascocarbs.
You're getting it. And these guys, they can be both beneficial and harmful. Think penicillium, the source of penicillin.
Oh, right, the antibiotic.
But then you've got things like claviceps, which can contaminate grains and cause ergot poisoning.
Not so good. And neurospora, I remember that one from the textbook. Used in research a lot.
Exactly. And let's not forget those delicious morels and truffles, ascomycetes too.
So next up, the Basidiomycetes. The ones that make mushrooms.
You got it. The classic mushroom shape, cap and gills.
But there are other types too, right?
Oh, yeah. Bracket fungi, puffballs, even rusts and smuts. Those are plant pathogens.
Oh, no fun. So all about Basidiospores this time.
Exactly. Formed on club-shaped structures called basidia, usually arranged in those fruiting bodies, the basidiocarps, what we know as mushrooms.
Makes sense.
And examples.
Well, agaricus, that's your common supermarket mushroom. But then you've got things like pustilago, a smut fungus that infects corn.
Yikes.
And puccinia, a rust fungus that goes after wheat. can be a real problem for agriculture.
I can imagine. So last but not least, what about those Deuteromycetes? The imperfect fungi.
Ah, the mysterious ones. We only know about their asexual reproduction using conidia.
So no sexual stage observed? Weird.
We haven't figured that part out yet. Many of them are saprophytes or parasites.
Any examples that come to mind?
Well, you've got alternaria, which is a common allergen, and then trichoderma, which is actually helpful, used in biocontrol to fight off plant diseases.
Fungi fighting fungi. It's a fungal world out there. But speaking of strange and mysterious, the book also mentions some entities that blur the line between living and non-living. Viruses, viroids, prions.
Now that sounds intriguing, but I think we'll have to save that for part 3 of our deep dive.
You're telling me? Those guys deserve their own dedicated exploration. We've covered a lot of ground with fungi already.
Okay, so we're back and ready to tackle these. Well, things that aren't quite alive, but kind of are. Viruses, viroids, prions.
They do make you think, that's for sure. What does it even mean to be alive? They push the boundaries.
So the book starts with viruses. A long history there. Dmitry Ivanovsky, back in 1892.
Ah, yes. Studying tobacco mosaic disease, found the infectious agent could pass through filters that trapped bacteria.
Meaning, whatever it was much smaller.
Right. And then Martinus Beijerinck, he took it a step further, showed that this infectious agent could reproduce, but only inside a host plant. Couldn't grow it on its own like bacteria.
So that's where the term virus comes from.
Yep, Latin for poison. Catchy, right. But it took a while to figure out what they actually were. Wendell Stanley, in 1935, he managed to crystallize the tobacco mosaic virus.
So what did they find? I remember the textbook saying it was a package of genetic material, either DNA or RNA, inside a protein coat.
You got it, a capsid, that protein coat.
Yeah.
And yeah, they can only replicate inside a living cell.
No cellular machinery of their own.
Nope, they're hijackers. They take over the host cell, use its resources to make more copies of themselves.
Like little molecular pirates. And of course, they cause a ton of diseases. Colds, flu, HIV, even some cancers, right?
Pretty much. And their simplicity is part of what makes them so effective. Small, streamlined, they can evade defenses and sneak right into the host's genetic machinery.
Now, the book also mentioned viroids, even smaller than viruses. No protein code at all, just RNA.
Naked RNA molecules, basically. But don't let that fool you, they can still cause a lot of damage, especially in plants. Disrupt growth and development.
Like little troublemakers.
Mm-hmm.
When were they discovered?
Theodore Diener, 1971. He was studying potato spindle tuber disease. That's when he found them.
Another piece of the puzzle. But okay, prions, those seem even weirder. They're just protein.
It's wild, right? No DNA, no RNA. For a long time, scientists were baffled. How can a protein replicate and cause disease without any genetic material?
Yeah, how does that even work?
Well, it turns out prions are misfolded versions of normal proteins found in the brain, and when a prion comes into contact with a normal protein, it can cause it to misfold too.
So one bad apple spoils the bunch.
Pretty much. And it creates a chain reaction. These abnormal proteins build up in the brain, and that's where the trouble starts. Disrupts normal function, leads to neurological disorders.
Like Creutzfeldt-Jakob disease.
That's one example. Mad cow disease in cattle, too. It shows how important protein folding is. That delicate balance is essential for cells to function properly.
Makes you think twice about those protein shakes. But okay, let's switch gears for a minute. Lichens, the book mentioned those too. A nice change of pace after all those disease-causing entities.
Absolutely, a breath of fresh air. Literally, they're all about cooperation, not destruction.
I always see them growing on rocks, trees, even buildings. They seem to survive anywhere.
They're incredibly resilient, and it's all thanks to that symbiotic partnership.
The fungus and the algae or cyanobacteria working together.
Exactly. Fungus provides the home, the protection, and the algae or cyanobacteria, they provide the food. Photosynthesis, just like with plants.
A perfect partnership. It lets them live in places where neither could survive alone.
Exactly. They break down rocks, help create soil, even act as bioindicators. They're sensitive to air pollution, so they can tell us a lot about the health of our environment.
Amazing. So we've covered a lot of ground in this deep dive, from the microscopic world of viruses and prions to those fascinating lichens.
A real whirlwind tour of life and the not quite life.
It really makes you wonder what else is out there, what other discoveries are waiting to be made.
That's the beauty of science. Always more to explore. And as we learn more, our understanding of life itself continues to evolve.
Well, this deep dive has certainly given me a lot to think about. Thanks for sharing your expertise.
My pleasure. Always happy to talk about the wonders of biology. keep exploring and stay curious.
That's it for today's deep dive. Thanks for listening.